So today I'm going to talk a little bit about modeling aging and specifically about neurodegenerative diseases and talk about some of our recent findings. So aging is the progressive decline of fitness with time. And as a result of age, there's an accumulation of damage and likely a programmed cellular response. In other words, it's likely that age is built into the systems. We don't know whether or not this is actually something that is pre-programmed or it's wear and tear that results in the deterioration with age. And it's a very hot topic of investigation. There are currently, as you can see on the left, a lot of things that happen with aging: genome instability, telomere attrition, epigenetic loss, drift, mitochondrial function, senescence. And if you go to an aging meeting, you often find that there are sessions which deal with each one of these individually. And our focus is beginning to be less about treating each one individually and more about looking at the interface between them, thinking of age and the consequences of aging in terms of their interactions between these systems. So what we do know, though, for sure, is that age is the largest risk for many diseases, not just neurodegenerative, but also psychiatric disease and cancer and diabetes. So I will focus on or make a lot of the comparisons between age-related degeneration and Alzheimer's disease. And we've shown previously that Alzheimer's disease is not just an extension of aging, but it is a disease itself. And I think that's an important distinction to be made. Now, as it stands now, although this hypothesis is being revised, has been sort of the amyloid hypothesis, where the tau tangles and amyloid plaques that exist in postmortem tissue clearly exist and are purely correlated with the disease, but it has been considered or hypothesized that it's causal or has been the initiating event. And it seems less likely given the last 10 years of 1,000 trials and $24 billion that have been spent on clinical trials that have failed, that this hypothesis needs to be revised and or elaborated. That being said, a lot of model systems have been used, but most of them are taking wild-type animals and overexpressing human mutations with the idea that they'd understand the disease. Now, it's not unreasonable to go in that direction, but as we're beginning to learn and appreciate even more all the time, is genetic background is a very important feature of any mutation that occurs. So it's not just the mutation itself, it's the background on which the mutation occurs that's important. We've moved to looking at human cells and taking into consideration the human genetic background as being an important feature. And with the discovery of the Yamanaka factors and the ability to reprogram, we've taken advantage of this. So initially, we took fibroblasts from patients and made fibroblasts and reprogram them to iPS cells. And when doing this, you can derive cells of many different lineages: cardiac, retinal, pancreatic. Really, it's amazing the number of cells that can be authentically be derived, including neurons. We did this study with 20 patients that ranged of age from 1 to 80 years old, so fibroblasts were young to old. We took primary fibroblasts and did RNA-seq, and we looked at the differences. We actually, using fibroblasts, we could find age-related disease, age-related genes that declined with age. Not a lot, but we did see some. And these were with early computational tools that have been advanced further since. However, when we looked at the iPS cells from the same isogenic patients, we found essentially no genes different between them. And then we took and made neurons from these iPS cells. We also found very little or no differences between with age, change with age. Well, what that means merely is that the iPS cells did their job, they reprogrammed, they erased the epigenetic program and turned them back into embryonic cells. And as we know, Alzheimer's disease, aging, or Alzheimer's disease really requires these age as a feature to be able to pick it. We've lost aging in this model. So we've turned to another model where we can use two genes, directly reprogram fibroblasts, NG2 and ASCII, which are small, which are pioneer transcription factors. And more recently, we've identified some small molecules that facilitate this direct induction. These pioneer factors open chromatin and allow for specific transcription factors to bind and induce a direct conversion of the cells. And this is just an area still of major investigation of trying to figure out exactly how these factors do it. But it's clear that they're opening chromatin for transcription factors to bind specific sites. But quite remarkably, in a 3-week period of time, we go from fibroblasts into this network of neurons. Now, when we look at the transcriptional differences between young and old, or across young and old, we now can pick up a number of genes that are significantly different. So we've preserved this aging phenotype, including one gene that we've spent a lot of time on, which is RAMVP17. It's a nuclear import gene that controls the transport of molecules from the outside of the nucleus to the inside of the nucleus, and is downregulated with age. And we developed an assay where we could monitor the nuclear import and export signaling ratio. And what we see is this nuclear import export is disrupted with age as a function of this gene. Now, the good news is that it's not a mutation in RAMVP that occurs with age, but rather an active downregulation, because when we make iPS cells out of very, very old fibroblasts that have that the fibroblasts also show defects in RAMVP17, we can reverse it by inducing iPS cells. So that's a reversible aging phenomenon. So since we've been doing these studies, we put all these vectors on Addgene and sent them out to many people. And a lot of people now have shown that there are epigenetic changes, methylation, neural survival, age-related changes that can be picked up now using this strategy that have not been previously found. Importantly, we think that aging, it reveals that aging is not just a single phenomenon, but certainly neuronal aging involves a variety of mechanisms. Here's one that we have done in collaboration with Steve Horvath, who developed the DNA methylation clock. And what you can see down here is this is chronological age here, and this is DNA methylation age here. And for iPS, human iPS cells, NPCs, and neurons, they are all chronologically aged around birth. So they're very, very immature. Alternatively, the IN fibroblasts and INs, if you look at their methylation clocks, they're all proportional to the age from which the fibroblasts, the age of the patient the fibroblasts were taken from. We can do this another way, which is to take the RNA sequencing clusters from BrainSpan, which are a set of genes that are expressed at various times prenatally and postnatally. And what you can do with this very useful tool is take your RNA-seq and feed it into BrainSpan, and it tells you how old your cells are. So what we find, for example, is if your cells were taken at 8-week, you can feed your cells and it'll tell you if they're young or old. And when we do this, what we find is that for the iPS cells, prenatally, they compare to prenatal, but they're low comparison similarity scores with age. And it's just reversed with our INs, where they're lower similarity scores prenatally and they get higher postnatally. Again, suggesting that they preserve their aging fate. So we did RNA-seq for all of these patients. And when we look at the fibroblasts from the control versus AD, we see perhaps as expected, but we were sort of hoping we might find some signature. differentiating AD from controls. We don't see any difference there. However, when we made IN cells, we have massive numbers of cells that differentiate between the age-matched controls and AD. So I'd like to make the argument that age is a crucial factor in being able to detect aging. It's a major risk factor, and all the genetic underpinnings can't be revealed until they're later. I'd also like to highlight the fact that what I've showed you is that it isn't one of these events that's important, but rather aging revealed metabolic problems, DNA problems, nuclear import problems, and it's really a combination of these. I think we think of brain aging and disease as really a homeostatic system, which is controlled by a variety of systems, all of which are compensating for each other in normal aging, but a secondary stress results in cognitive decline. And that sort of description should give you a hint to this idea of cancer, where you have age-related decline or age-related changes that occur with cancer, but a second hit. And you can compensate with, even if you have a genetic background for cancer, but if you have a second hit, which makes you more vulnerable, then you can have cancer, just like we believe you can elicit cognitive decline. All right, so let's go a little bit deeper and try to analyze more carefully and more systematically the changes that are occurring in age in not just our cells, but in postmortem tissue. So we used WGCNA to find clusters of genes from our transcription from the AD patient INs, and we pulled out clusters, or we call AD modules, that are clustering around that give an insight into those events or clusters of genes that are involved in upregulation or downregulation of the cognitive decline that occurs with aging. And we did the same thing for postmortem tissue. So we took existing data from postmortem tissue of Alzheimer's disease, performed the same WGCNA, and what we find in both cases is a standout motifs that emerge is a metabolic shift to glycolytic reprogramming is indicated both in the IN and from this ROS map. And what we find is the highest order difference that describes the differences between AD in both of these data sets is a single gene called PKM, or pyruvate kinase. And it's the difference between these two is a single splice variation. So the PKM2 has an extra exon called exon 10. So you can distinguish between these two. And while the expression of PKM is not different, total difference, there's a clear difference in the ratio of PKM2 to PKM1. Now we have 633 transcriptomes, and what we see, and we can now map these 263 to their Brock stage, and we find that PKM1 isoform downregulates with more severe Brock staging, whereas PKM2 is upregulated. So again, indicating that this PKM2 isoform switch is correlated with diagnosis of Alzheimer's and severity. If we dig down a little bit deeper into this and look at genes that are associated with the PKM2 version, where you have HIF1 alpha activated, STAT3 activated, and glycolysis genes more heavily activated, we see a serious upregulation of these genes at this point. And this is reflected here in this heat map, where you have an increase in these genes in neurons. So what happens normally is that PKM2 is kept cytoplasmically, where it can provide the pyruvate it needs to feed into the oxidative phosphorylation pathway. Alternatively, the PKM2 is phosphorylated and transferred into the nucleus. There, it co-binds with HIF alpha and enhances the binding of HIF alpha, HIF1 alpha, and STAT3, and induces a severe stress and pro-apoptotic genes, and further amplifies this glycolytic pathway. Now, to prove that in our cells this is happening, we can see that even in our neurons, we're getting a shift of the cytoplasmic PKM2 into the nucleus. And we can look, let me get you out of the way here. We get this downstream target gene, which is H3T11, which is a PKM nuclear target gene. So what's in the nucleus, you do see this upregulation, and we see it too. So it's not just transferring this protein to the nucleus, it's upregulating many of these Warburg-like genes. If we then do ATAC-seq on this data and look at the chromatin around these targeted PKM2 targeted genes, like HIF1 alpha, STAT3, CT, and MV1, they all have, well, these two have significant chromatin opening relative to AD relative controls. No different in this one, but these two clearly are showing open chromatin as well. So there's a increased chromatin accessibility for HIF1 alpha also as part of the gene program that's involved. So what can we do? What can we do to protect these cells against that? Well, because this is such a well-known target in cancer, there are drugs being developed for cancer for shifting PKM2 to PKM1. And one of them is a natural product called chiconin. And this wonder drug forces the dimer PKM2 to heterodimerize. And when it heterodimerizes, the theory goes, it won't be able to get into the nucleus, it'll stay in the cytoplasm, and it won't activate HIF1 alpha or STAT3 and get that whole pathway going. So we did this, treated our cells with chiconin, and the first thing you can see is this cobalt chloride induced nuclear localization is blocked. So we can prevent the influx of PKM2 to the nucleus, and we can reverse much of the metabolic dysfunction. So our lactate pyruvate ratio is back to normal. This stumbling block here at the 1,3-bis glycerol is not affected. And then when we go further to look at RNA-seq on the AD controls in the presence of chiconin, whereas normally we see the ADs showing a very nice shift difference from the controls, these arrows indicate the RNA-seq directionality shift that occurs with chiconin treatment. So chiconin induces a transcriptome shift in AD, and it's back to their more control-like state. Remarkably, when we then go and look at the RNA factors that we found that were downregulated in AD, that is synaptic transmission, synaptic plasticity, long-term potentiation, those are reversed also in part by this treatment with chiconin. With AD compared to the controls, which is this pink, there's sort of a de-differentiation. The cells don't fully mature, but with chiconin in the same AD patients, we're getting a much more mature phenotype or trajectory of differentiation. So to summarize here at the end, there's a cancer-like PKM2 that it's upregulated. The splice form variation that occurs in AD is causes a metabolic shift and a fate loss. And the cells in AD become vulnerable, resulting in loss of function in terms of metabolic activity and gain of function epigenetically. There's an exchange of profile, and this looks very much like cancer in the sense of the changes that we're seeing. One thing I'd like to point out to you, if you look at this chart here, these are how different cells in the central nervous system use different metabolic activity. So from proliferation, glycolysis, metabolic flexibility, anabolic pathways, neurons are unique in their dependence on oxidative phosphorylation for generating most of their ATP. Other cells share glycolysis and other fates and their ability to generate energy. But this also means that AD neurons cannot easily use glycolysis and the phosphate shunt pathway to develop to generate more nucleotides or amino acids or lipids that they may need in the damaged case. So I think what this tells me is that neurons are particularly vulnerable to damage because they don't have this flexibility in energy metabolism, and they're so dependent on oxidative phosphorylation. They're fine as long as everything is healthy, but as we age, it makes neurons more vulnerable than any of the other cells in the brain.